Hey, welcome to lesson two of this course. So, we're going to be talking a little bit about microscopes, why they're useful, and also how we distinguish different microorganisms under the microscope. So, a brief history of microscopes. We touched on a little bit with Antoine von Leeuwenhoek, who was the first to see living microbes with a very simple microscope. So he had just what is a single lens microscope. Think of it as very powerful glasses, essentially. And then after him came along Robert Hooke. So he coined the term cell because under his microscope, he was able to see in cork, which is the bark of a certain tree, a cork tree, that there were these little individual units with which he likened to the cells that monastic groups would live in because they're just small and next to each other. Now, he had what we call a compound microscope. Compound microscopes have two or more lenses. So these are the two people that really led from the legacy origin of microscopy, of just being able to see cells and kind of call out this is a cell. This is a brand new thing, and we don't really know how to talk about it, calling it a cell after uh, monk living quarters and turning it into what is modern day, very intensive, very detailed microscopy. So we're going to talk a little bit about how microscopy works in general. And usually it comes down to the light source, the lenses you're using, or the type of receiver that you're using. In a normal compound microscope, you shine a light through a sample that goes into the lenses, and you put your eye over the lenses and you're able to visually see. But some types of microscopy actually use sensors. We're going to really be focused on compound microscopy for this portion of the course. So just know that your eye is ultimately the sensor that you are looking at these microorganisms under. A couple quick key vocabulary words. So there's magnification and then there's resolution. Magnification is going to give you the size of the image. So if you think about something really, really small, you can't see it with the naked eye, you need to magnify it. You need to make it bigger. But as you make it bigger, parts of it might be fuzzy. So you need to increase the resolution or the clarity of that to distinguish different parts so you don't get a fuzzy image. So magnification, make something bigger. Resolution, be able to see the details of that image. If you have glasses, you take your glasses off, everything's fuzzy. So glasses actually are increasing the resolution of things, not the magnification. Binoculars would increase the magnification. The other thing is contrast. So you're looking at something, perhaps it is very dark on a light background or very light on a dark background. That's fantastic. But sometimes things look very similar, have the same overall, what we call a refraction index. So when light travels through something, it bends, and the amount of bend is determined by the refraction index. So if you're looking through glasses, how these work is you're refracting all the light to specific points, and that is different for everyone's eye. That's why everyone has a different glasses prescription because you're refracting light in different ways. Glass does that. If you've ever, looked underwater, the refraction into the water is going to be different than through air. So those are two mediums that light passes through. Through air, it's going like this, and then it hits water, and then it will bend because of that refraction index. And when you have things with the same overall contrast and refraction, it's really difficult to see. But one thing we want is the substrate that a microorganism is in to match the refraction of the glass that we're using on slides. So usually you'll take a glass slide on the bottom because you need to be able to shine light through it, and then you'll put the microorganism on the slide in a medium, and then you'll put another glass slide on top. and you want whatever that medium is in between those two slides to match the slides. So this is usually going to be something we call immersion oil, which matches that refraction index. And this is all to get a very clear image going through the organism, whatever's sitting there, and direct that light back through all the lenses into your eye. Now, like I was saying, contrast is a huge thing. So there's different microscopes that we'll use for different kinds of organisms. So bright field microscope uses a dark specimen on a bright background. So these are very common in stained slides. There's also a dark field way of doing microscopy where you have a bright set specimen on a dark background. This is usually used for things like live cells and determining uh, motility. So if the cells are moving around, you want to be able to see those very clearly so you can track them. So you might use a dark field microscope for this. There's phase contrast, which enhances the internal contrast in live cells so you can see those interior structures. So if we think back and think about the naming of cells. They're called cells because they look like these uniform things. Well, with that original microscope, they weren't able to see all the organelles inside of the cell. So that's why phase contrast can be really important. Now, I briefly have to explain antibodies to explain fluorescence and getting into electron microscopy and things like that. So, antibodies, if you're not aware, are these Y-shaped proteins. And very briefly, they have this Y shape, and at the head, they have something called the recognition area, is the term I'll use. So, that tip of the antibody recognizes certain parts of protein. So you can take a protein, think about a very common protein would be something on a blood cell. So you have a bunch of cells in your body. Red blood cells are those little platelet cells. On that, there's a bunch of different proteins. We can have an antibody specific to a protein on a blood cell. And then on the other end of that Y, down here at the base, we can put something that is fluorescent. Now, if you've ever seen any of those videos of beaches that have the bioluminescence, that's kind of what we're talking about here. It's a protein that gives off light by itself when you feed it certain substrates. So by taking an antibody that's specific to part of a cell, let's say the nucleus in a eukaryotic cell is going to attach to the nucleus, and giving it a fluorescent tag, we can actually mix the antibody with our cells and then have a very clear image of where the nucleuses are in all these different cells. And this might be important. You know, you have cells dividing. Part of that is the nucleus is undergoing a lot of different changes, and we want to watch those changes over time. So we're going to use this antibody fluorescent tag. Now, um, there's also indirect and direct immunofluorescence. The textbook goes into more detail on that specifically. It's a little bit difficult to wrap your head around for the first time. So if you have any questions, of course, just message me. Now, the other thing we're going to talk about is electron microscopy. And this is really, really cool, but kind of, again, difficult to wrap your head around. So instead of using your eye, you're going to use a sensor. There's going to be a sensor at the top, and you're going to look for radiation. Instead of light. So radiation is light, is just in a wavelength that we can't physically see, and that's why you need the sensor. So you're going to take something like a cell, you're going to put it in a medium in between that slide that is a medium that contains radioactive isotopes, and then you're going to put that under a electron microscope, and the electrons coming off of the radioactive isotope will actually show you the structure very, very highly detailed of a cell, or in most cases, actually a virus. So remember, viruses are a lot smaller than cells, and that means they're very difficult to see. You can't really do it with a typical compound light microscope. So those very common microscopes that you probably used in high school. Instead, you have to use either TEM or SEM to see the detailed structures of these viruses. Now, TM shows you these internal structures at around 1 to 10 nanomolar resolution. It depends on the microscope and a lot of other details that I'm not going to get into, but you get highly detailed information. And SEM will give you a very detailed surface topology of what that virus looks like. So TEM is kind of this flat plane of whatever you're looking at, similar to a typical microscope. SEM, you can actually get detailed 3D surface models from. So these are very, very cool. I've used TEM a lot, and that is always to look at viruses when we use it. Now, I also will say, again, this is where you're going to have to go into the textbook and the lesson a little bit to get the details because it is a little bit more complicated. And again, just ask me any questions that you have. Now, we're going to be talking about staining. So, why would you stain something? Again, this is to increase contrast. Most bacteria are going to be colorless. A lot of other archaea are colorless. So, you need to stain, and that adds the contrast so that you can see what is actually going on. There is what we call simple staining, which is just one dye. So, that might be for the whole cell. It stains the whole cell, all the different internal structures are stained as well along with that, and then everything else is unstained. Or it can be a differential stain. So you can have a stain that's specific to a certain type of bacteria, to certain structures in the bacteria, and that will again inform the taxonomy, that classification system of what this organism is. So if you're sitting in a clinic and saying, I have someone who's really sick, I took a sample, I put it on a slide, and I need to know if it is gram positive or gram negative bacteria. So the gram positive bacteria, or gram negative bacteria, they have um, no chromophore. So that is a just part of the cell wall. If they're negative, they don't have that. If they do have it, you can stain for it. So they're chromophore positive or gram positive bacteria. So when you're looking at it, you can immediately classify with this stain, is it gram positive or is it gram negative? Maybe the antibiotics are different for both of those. So that's why that is important. Um, Getting a little bit more into it, when you're doing these gram stains, which is one of the most common stains that you're going to do with bacteria, you have multiple steps. So there's crystal violet, which is the primary stain, there's iodine, there's alcohol to decolorize, and then there's Safranin, yes, that's the word, to counter stain. So when you have gram positive, it's going to come out as this purple with thick walls, and then gram negative is going to look a pinkish red. The outer membrane is going to actually be washed off with alcohol, and then it's going to take on that safranin color, which is that pink red. And this is very, again, rapid guidance for clinicians in a hospital or primary care or a field setting where you can say, okay, we have something here that is gram positive. We have antibiotics that work on the majority of gram positive bacteria. So we're going to give the patient that. And then we're going to do further investigations to figure out the exact bacteria that it is. And that's why this is such a powerful and important stain to understand. Now, another very powerful stain is acid-fast staining. So this is actually for Mycobacterium. So something like Mycobacterium tuberculosis, TB, that's a mycobacterium. So someone comes in with a cough, you take a sputum sample, you stain it, and you do an acid-fast stain. So this shows that it is red, and then you can just say, okay, the acid-fast stain popped up red. That means that this is some sort of mycobacterium. Most likely, it's going to be tuberculosis. So, you're going to use that as part of your diagnostic. C. Endospore staining is another one. So, that's for Bacillus and Clostridiums. So, spores are essentially part of the life cycle of a bacteria. So when you expose bacteria to extreme temperatures or extreme conditions in general, they'll form these little sporelings. So it's even smaller than a bacteria, and that spore will wait until it has optimal conditions to then revitalize itself. This also happens with funguses and other organisms. It's a very, very easy way for bacteria to have their progeny survive over time when conditions become too disruptive for their normal life cycle. With this, you're going to have spores turn out green, so you get a green stain when it has spores, and if it's a cell undergoing its normal life cycle, it'll turn out pink. That pink again comes from the safranin that you're staining cells with. Clinically, things like anthrax fall into here, tetanus, botulism, and a lot of other environmentally stable spores that when it gets into your body is going to start replicating and be an extremely deadly disease. So one example of this is actually valley fever. If you've lived long enough in the Southwest, you've probably heard of valley fever and know that it can be a very dangerous, debilitating disease. And this is one of the ways that it could be detected. There's also capsule staining. So the background is actually stained, and a capsule will form this halo around cells. So this is a way to show the, I guess, number of capsules that you're dealing with, and it can be useful from the point of view of you're doing like more cell counting sort of thing. So how many bacteria really are there? It might be difficult if you do a normal stain to go through and count all the bacteria, especially if there's a lot. With a capsule stain, it might be a little bit easier. And lastly, there's a flagellin stain. So flagella are used, they're little tails that are used for motility of bacteria. The bacteria swimming around wherever they are, whether it's in a pond or in your bloodstream. That's how they get around. This will show the presence and arrangement of the flagella, which again is used for that taxonomic coding of which bacteria is this, what do we give the patient when they're infected with this type of bacteria. So you do, say you're gram negative, gram positive stain, okay, you know it's gram negative. Then you do a flagella stain, you see that it has a specific type of flagella that puts it in this subcategory of gram negative bacteria. And for that whole subcategory, there's an antibiotic that's very specific that works very well. You give that patient that antibiotic. Very simple, very straightforward. So really, you're combining two pretty simple but powerful technologies when you're doing work like this. You have that compound microscope, and then you have all these different stains that you can do. The microscope will, of course, be dependent on the specimen. There's a lot of details here, like is it live? Is it fixed? Are you looking at the surface? Are you looking at the internal structures? Are you wanting to, see if it's a fungus versus a bacteria? Well, that's very simple. You can just look at that without a stain. You don't need a stain to tell the difference between a fungus and a bacteria under a microscope. But if it's gram negative versus gram positive, then you have to get into staining. So these are things that layer on top of each other that ultimately lead to the type of specimen, the type of microorganism that you're looking at. You understand what that is, and then usually you're either going to do science on that specific microorganism or you're going to treat a patient that's infected with that specific microorganism. Another example is water treatment. So, testing the water, seeing if there's certain bacteria in it, there's certain different contaminants in there. You can do all that at the same time under a microscope. When it comes to looking for specific bacteria, maybe you're trying to determine, well, we had a dam break and it's all this runoff that really is getting everywhere in the city's water system. We need to determine if the treatment that we're doing currently is getting rid of all that new bacteria and maybe even new types of bacteria that are coming in. So in Arizona, of course, we have all the monsoons. The water treatment plants are designed to deal with all that excess water getting into their systems and making sure that we're not drinking bacteria that washes off of our roads into drains that then go into that reprocessing. So it's really important to understand when to use specific types of microscopy and then also when to layer these stains on top. Um, when it comes to things, the more complicated things like fluorescent microscopy, then you're getting into looking at specific structures, specific proteins. That's going to be more on the scientific side. So maybe you're trying to identify if this is actually a new species. It looks exactly the same under all the stains as another species of bacteria. And then you do some genetic work and you do some probing with those fluorescents to show there are structures in this bacteria that are different than this bacteria. And similarly with TEM and SEM, you're looking at very detailed structures on, say, viruses. So maybe you're looking at two viruses. They look very, very similar under TEM because you're taking cross sections. You're looking at just the cross section spot. And then you look at under SEM, you get that more detailed 3D structure and you say, oh, okay, this is actually something very different. So it's all about differentiating things at the end of the day, and then using that either to inform your science or to inform your clinical treatment of patients. All right, I think that is about it for this lesson. Again, if you have any questions, please feel free to message me and I'll clarify it as much as I can. Of course, there's a lot more detail in this than what I went over in this video, but hopefully this is a really good starting point and now you understand why these are necessary steps to take, especially in the clinic.